Hi, everyone. Thank you so much for joining us today. My name is Christina Asa. I'm a scientific application specialist here at Fujifilm VisualSonics, based out of our office in Toronto. I'm very excited to see such a large number of people joining us today. I know that there's still people trickling in, but we're going to get started as it is the top of the hour. So today we have a really exciting presentation entitled Measuring Vascular Stiffness in Aging Mice Using High-Resolution Vascular Ultrasound. And our presenter today is Dr. Kirsty Foote. To share a little bit about our presenter, Kirsty Foote gained her PhD from the University of Glasgow in 2012, investigating the role of a transcription factor in the heart during myocardial infarction. This work was funded by a capacity building award from the BBSRC designed to promote in vivo scientists in the UK. This became the starting point for her training in a wide range of surgical models and in vivo instrumentation for functionally characterizing clinically relevant animal disease models. Her first postdoctoral position at University College London investigated mechanisms of attenuating calcium dysregulation in the heart during ischemia reperfusion injury. Dr. Foote moved to the University of Cambridge in 2014, where her research broadened to systemic vascular disease and in particular, vascular aging. Her current research focuses on the functional alterations of aging in the vasculature and the molecular mechanisms that underpin the vascular aging process, with the overall aim of identifying potential therapies for slowing aging and delaying the onset of age-related cardiovascular diseases. So without any further ado, I'd like to pass things over to Dr. Kirsty Foote.
Thank you very much for that kind introduction, and thank you all for being here today joining us for this webinar. As mentioned in the introduction, my name is Kirsty Foote and I'm based at the University of Cambridge in the UK. And today I'll be talking about measuring vascular stiffness in aging mice using high-resolution vascular ultrasound. This is the outline of the presentation. I'll begin by giving a brief introduction to vascular aging in terms of its definition and relevance. I'll then go on to talk about why it's important to be able to measure vascular aging in small animal research. I'll then describe how we can use ultra-high frequency ultrasound to assess vascular aging in mice, and then describe how we apply vascular ultrasound in our studies to mouse models of delayed vascular aging. And finally, I'll wrap up with any closing remarks and discussion. You can track the progress of the presentation using the toolbar at the bottom of each slide. So I'd like to begin with a very famous quote, "A man is as old as his arteries." This was a phrase used by the English physician Thomas Sydenham in the 17th century. And this idea that the arteries age as we age and that this can vary between individuals is not something that's new and has been known for centuries. However, the research in this area is still relatively new. When we talk about vascular aging, we're referring to all the cumulative changes that take place in the vasculature with normal age, and this takes place in the absence of other diseases and begins in utero. As many of you will know, but just to give you an idea of the scale of the problem of vascular aging, if we take a look at the traditional risk factors that heighten our risk of developing cardiovascular disease, these are things like hypertension, smoking, cholesterol, and diabetes. All of these are going to increase our risk of developing CVD. But if we look at what happens with the effects of aging, you can see that this substantially increases the risk and accounts for more than 90% of cardiovascular deaths each year. And so it is important as it's the leading risk factor for developing cardiovascular disease. Vascular aging, this happens because there are changes that take place in the blood vessel with age that can then manifest and lead to other vascular complications such as cerebral infarcts, myocardial infarction, chronic renal disease, aortic aneurysms, and atherosclerosis. So it makes sense to try to target or potentially research vascular aging itself as a way to try and combat the onset of age-related vascular diseases. So what are the changes that take place in the aged artery? Many of you will be familiar with these, but the aged vessel typically has a thickened vessel wall, and this is due to proliferation and migration of the vascular smooth muscle cells, which make up the bulk of the vessel wall. There is often fragmenting of the elastin fibers and increases in collagen deposition, and both of these together lead to an increase in vascular stiffness. There is also dysfunction of the endothelial cells and often a low-grade chronic inflammation present. If we take a step closer or a step deeper, what we're really interested in is what's happening at the cellular and molecular level. And thanks to recent advances in aging research, we now know that there are a number of distinct processes that are characteristic or hallmarks of the aged vascular cell. And I've outlined some of these here. So these include things like damage to the macromolecules of the cell, genomic instability, damage to the mitochondria. There's often high levels of oxidative stress and presence of reactive oxygen species. The telomeres at the end of the chromosomes become shorter, and this can affect the lifespan of the cell. And there's an increase in what's known as inflammaging, or this chronic low-grade inflammation, and imbalances in things like proteostasis. So thanks to the identification of these distinct processes in the last decade or so, came about the advent of what's known as the field of neuroscience. And this looks to target the primary aging process rather than the secondary diseases. And what this involves is manipulating the underlying biology of aging, and therefore could targeting any one of these molecular processes potentially slow aging. And by doing this, we can then deduce if this is cause or consequence of aging, which is informative for future therapies with the overall goal of potentially slowing aging and the onset of age-related vascular diseases. And this brings me on to the importance of mouse models in vascular aging research because of the invaluable possibility to be able to genetically modify mouse models to generate new models. So we can genetically modify these animals to manipulate these hallmarks of aging and see whether this has any direct effect on the rate of aging. And this is particularly important for tissue-specific manipulation, so vascular-specific genetic modifications, which allow us to then directly see the effects on the tissue. And of course, this wouldn't be possible in humans. The other advantages of using mice are that the phenotype of vascular aging in mice recapitulates that of humans. And also they have a very much shorter lifespan. So this has got lots of advantages in terms of time and cost for research studies. So the strategy here would be to genetically modify an aging hallmark in mice and then assess physiologically whether this slows their vascular aging. And the question then is, how can we do this? How can we assess physiologically or in vivo mouse vascular aging? The way we do this is by measuring the hallmark of vascular aging, which is vascular stiffness. So as I mentioned in a previous slide, vascular stiffness is mainly caused by changes to the collagen levels and the elastin integrity in the vessel wall. And what we can do to measure this is by measuring what's called the pulse wave velocity. So when the heart beats, it generates a pulse wave or a pressure wave that transmits through the arterial tree. And the speed at which this pulse wave travels from one part of the artery to the other is directly affected by the stiffness of the vessel wall. So in a younger, healthier vessel, where the walls are more elastic and supple, this propagation velocity will be slower than, say, that of an aged vessel, in which the walls move less and there's less resistance to the propagation wave. And so a faster pulse wave velocity would be consistent with an aged vessel or a stiffer vessel. And the way that we can calculate pulse wave velocity is, of course, using the distance between 2 points along the artery divided by the transit time for that wave to travel from one point to the other. And this is the gold standard method used in the clinics, usually using carotid to femoral technology. We can also look at vascular stiffness by looking at the biomechanics of the vessel wall. So this is the ability for the vessel wall to expand and recoil and is a function of its volume and pressure changes. There are several parameters that describe vascular stiffness, including arterial compliance, which is the ability of the arterial wall to increase in volume with increasing pressure. You've got beta stiffness index, which is the ratio of change in pressure to relative change in diameter, and arterial distensibility, which is the relative change in diameter or area for a given pressure change. So these parameters are quite similar, but they tell us slightly different things about the vessel biomechanics. And they rely on measurements of volume or diameters of the vessel and pressures as well. So how do we do this in small animals? So if we take the formula for arterial compliance, which, as I mentioned, is the change in volume for change in pressure. So what we can do is we can use ultrasound for measuring changes in volume or diameter. And as many of you will know, ultrasound is a non-invasive technique which emits ultra-high frequency ultrasound waves through the skin into the body, and the reflected waves will build up an image of the underlying organ or vessel. So in the case of vascular ultrasound, you'll see something that looks like this. And also in combination with blood pressure measured by blood pressure cuff or intra-arterial blood pressure measurements, together this can give us the numbers that we need to plug into the formula and give us the data for indices of arterial stiffness. So in terms of ultrasound, and this is what we use in our lab, we use the VisualSonics Vivo 3100 system, which is a great machine and can offer lots and lots of different benefits for aging or vascular research. The main advantage is that it's non-invasive, which is very good for aging and longitudinal studies where you want to track vascular changes over time in the same animal. You get real-time detection. There's ultra-high frequency up to 70 megahertz, and this allows for a very high resolution down to 30 microns, which is very good for imaging very tiny blood vessels. Another excellent option that you can do with this is measure the pulse wave velocity, which I'll go on to explain how we can do that using ultrasound. And we can also detect aneurysms and perform 3D reconstructions. You get lots of different vessel wall characteristics such as diameter, strain, velocity, and stiffness. And we can also quantify blood flow. So this is the way in which we do this in mice for our studies. So the animal is firstly anaesthetized with isoflurane and positioned in a supine position on a heated platform, as shown here. The paws of the animal are taped down to these electrodes, which allow us to measure the heart rate and the ECG. We monitor body temperature using a rectal probe, and we can also supplement any heat with a heat lamp if necessary. And then we remove the fur from the whole neck region in preparation for imaging of the carotid artery. We then apply these gauze rolls either side of the animal, and this allows us to apply ultrasound gel and prevent it from slipping off the animal during recording. And I've got some tips and tricks for anyone that's looking to improve their methodology of this, or they're starting out. These are some of the things that we've found have markedly helped with our imaging quality and consistency between experiments. So, for example, effectively removing the hair is important. So we would always recommend a depilatory cream rather than a shaver. We do supplement the heat to the animal if necessary. The platform is heated, but we like to keep the animals within a strict temperature range. And sometimes heat lamps or gauze coverings can really help. We always warm the gels, the ultrasound gels, which helps to prevent bubbles, and always make sure to use enough. That always helps to improve image quality as well. And as I mentioned, we use these gauze rolls, which are just little squares of gauze rolled up and taped to act as a way to stop the gel slipping off the animal. And we also make use of the angling and the tilting that's available on the platform and with the probe to maximize image quality. So we begin recording in the short axis mode. So this is where the long length of the probe is perpendicular to the length of the animal. And this is an example of an image that we would get. So we do short axis first in order to identify the structures in the neck. So you can see here you've got the trachea in the middle, and then either side you've got the carotid arteries. And you'll see that on the right-hand side carotid artery, there's two openings, and this is representing the bifurcation of the carotid, which we're looking to focus on because it helps us orientate ourselves. And then once we've secured the short axis, we then position the right carotid artery into the center of the view, which is what we are interested in. And we then rotate the probe 90 degrees so that the long axis of the probe is parallel with the long length of the animal. And we now can see our carotid artery in this view. And you can clearly see the lumen, the walls, and the bifurcation. And then, as I mentioned previously, we do ultrasound imaging, and we also do blood pressure measurements. So this is an example of our layout in the animal facility, where we've got ultrasound imaging going on one side of the room, and then we've got a surgical setup on the same bench. And to describe our workflow in a bit more detail, so once we've got our animal anesthetized, we begin with carotid artery ultrasound, as I mentioned, and that allows us to generate indices of diameters from an M mode, and allows us to do various different analyses, including speckle tracking. And once we've imaged the carotid artery, we then move the animal across for surgery. So under the same anesthesia, we would then surgically isolate the same carotid artery and expose it for blood pressure measurements. So for this, we use a blood pressure catheter, which is a very thin hair-like wire with a pressure transducer on the end of it. And this is designed to fit inside the carotid artery, where we can generate real-time blood pressure measurements. And then the third part of our experiments involves measuring aortic pulse wave velocity. We do this invasively and non-invasively. Invasively, it requires the use of, again, another blood pressure catheter, but instead of a single pressure transducer, it has two sensors separated by a known distance. And this catheter is inserted into the femoral artery and advanced into the aorta, so that you have one sensor in the thoracic part of the aorta and the lower sensor in the abdominal part of the aorta, where you get blood pressure traces that look like this. And knowing the distance between the sensors, which is always fixed, we can then divide by the transit time, and that will give us a pulse wave velocity. In this case, it's 3 meters per second, as an example. But as I mentioned, we can also measure pulse wave velocity non-invasively. And this is one of the benefits of the VisualSonics Vivo VASC software. So in this case, I've given an example of the carotid artery, but you could also do larger arteries like the aorta. So this involves recording what's called an EKV file. So this stands for ECG gated kilohertz visualization. And this basically allows greater frame rate files. So this is inputted into the Vivo VASC software. You can then draw a region of interest around the vessel, or you can even just draw it around the upper wall. And then you will get images that look like this, showing the movement of the vessel wall from the left and right side of the region of interest. And then by placing the cursors at the base of one inflection and then the base of the second inflection, it will then tell you the distance between the two and the transit time. And then from that, you can work out the pulse wave velocity or the pulse propagation velocity. And you can see that in this case, we were getting numbers very similar to the invasive method. Other things you can do with the ultrasound in terms of quantifying vascular function is measuring the Doppler blood velocity. So this relies on the Doppler principle. So what this means is that there can be a change in frequency from the outgoing wave to the returning wave due to the movement of the blood cells. So the formula for Doppler frequency is as follows here, and takes into account things like the transmitted frequency, the speed of sound in tissue, and the Doppler frequency shift is proportional to the blood velocity. And it looks something like this, where you can then place cursors and measure systolic and diastolic blood velocity. There's also a Doppler angle correction, which takes into consideration the angle at which you scan the vessel, which should be below 60 degrees to avoid error. And also just a note that it's good practice to keep the physiological parameters within a strict range as much as possible. So this gives you measures of heart rate, respiration rate, and body temperature. The other features that we measure to quantify vessel properties are things like displacement, strain, and intimal medial thickness. And this is all available on the VivoVasc software. Again, where you input an image file or your EKV file, and then you can draw regions of interest and then generate the data. So putting this into context of our work, so we've used this technology to measure vascular stiffness in aged mice. We first investigated when this happens in wild type C57 black 6 mice. We wanted to test a range of time points to understand when the onset of vascular aging occurs. So we used multiple time points and we applied this technology, and what we found was that quite consistently across most of the vascular stiffness parameters was that the change would occur by 44 weeks of age. So we used time points of 8, 22, 44, and 72 weeks. And we found that quite consistently we were seeing the changes occurring between 22 and 44 weeks of age, with reductions in arterial compliance, an increase in stiffness index, decreases in arterial distensibility, and increases in pulse wave velocity, all of which are consistent with vascular stiffness. And so we were able to show for the first time the age window at which the onset of vascular aging in mice begins. And we also looked at structural data from the vessels from these mice and we found that there was consistencies with the structural changes that occur in vascular stiffness as well. So for example, the collagen levels were found to be significantly increased by 44 weeks of age. And similarly, the number of elastin breaks were also found to be significantly increased by 44 weeks of age. So this was in support of the in vivo vascular stiffness data that was seen to occur by 44 weeks of age in wild-type mice. So as I mentioned earlier in the presentation, we're interested in potentially manipulating molecular mechanisms of aging and seeing how that can affect the rate of vascular aging in mice. So we're particularly interested in genomic instability, and we use mouse models to investigate this. So we're particularly interested in a particular type of DNA damage known as oxidative DNA damage. And this is where there is damage to DNA bases by reactive oxygen species. And this causes replication stalling and mutations. And the most frequent oxidative DNA damage is known as 8-oxoguanine. And what happens during oxidative DNA damage is that a guanine base is oxidized and becomes oxoguanine. And this is then replicated, and the problem here is that oxoguanine can functionally mimic a thymine, and this can lead to mispairing and mutations. Fortunately, the cell is equipped with repair mechanisms to tackle this type of damage, and the major enzyme that's involved in this type of repair is known as oxoguanine glycosylase 1, or OGG1. So this works by base excision repair. It binds to DNA, excises out the wrong base and replaces it with the correct base, corrects the mutation. In terms of what's known about oxidative DNA damage and its association with aging, it is associated with aging, we know this, but it's still not fully understood. We know that oxidative damage is present with age in some studies, but not others. And it's unclear whether it's a cause or a consequence of aging. Therefore, our study hypothesis was that if we enhanced base excision repair through genetic manipulation of OGG1, that this could reduce oxidative DNA damage and delay vascular aging. And this is what we've done. So we've manipulated the major enzyme for base excision repair, OGG1. And so we have mice with a vascular smooth muscle cell specific overexpression of OGG1. So under the promoter SM22A, which is for vascular smooth muscle cells, these mice have, this is a model of enhanced base excision repair. We've also got a vascular smooth muscle cell specific inactive OGG1 mutant. So these animals do express OGG1, but an inactive mutant form. And this is a model of defective base excision repair. And we've compared the rate of vascular aging in these mice with OGG1 nulls, which are also a model of defective base excision repair, and wild type controls. And we age them through the same aging protocol as described previously. And for today, I'll focus on the time points from 22 weeks onwards. So again, using our methodology as described, the combination of ultrasound with blood pressure, we've measured vascular aging in these animals. And what we found was that at a young time point of 22 weeks, we saw an unexpected but transient protective effect in the null animals, with an increase in arterial compliance and a reduction in pulse pressure, which wasn't expected given that they have no OGG1 and potentially higher levels of oxidative DNA damage. But as we aged the animals further, we noticed that the animals which had defective or null OGG1, that's these ones and these ones, had worse vascular function. They had higher pulse wave velocity, consistent with increased vascular stiffness. And when we got to 72 weeks of age, again, if we look at pulse pressure, we can see that the animals with null or defective OGG1 had higher pulse pressures compared to wild type, which is consistent with increased vascular stiffness. And in terms of arterial compliance, the animals with the overexpression of OGG1 were performing better than all the other strains. And then when we show the data all together, this is showing each animal strain and how their own aging transition occurs. So each animal's been normalized to its own 22-week time point. And what we can see is across the board, across all our parameters, is that the rate of vascular decline is faster in the animals with null or defective OGG1, whereas the animals with the overexpression had very little change in vascular stiffness as they aged, highlighting a potentially protective effect of OGG1 with age. So just to summarize the presentation and the take-home messages, vascular aging is the leading risk factor for cardiovascular disease, and therefore there's a critical need for being able to precisely quantify vascular aging in mouse models of vascular aging in vivo. Ultra-high frequency ultrasound allows for reproducible quantification of vascular stiffness in small rodents, allowing us to generate multiple standardized parameters. And using ultra-high frequency ultrasound, we show for the first time that mice develop arterial aging by 44 weeks of age, effectively delineating the onset of vascular aging in mice, which is informative for the aging community, allowing for potentially shorter working protocols. And in our applied mouse model, this was mice with null or defective DNA repair enzyme OGG1 showed increased vascular stiffness, whereas overexpressing OGG1 protected mice from vessel aging, highlighting the sensitivity of the technology that they were using to be able to detect changes resulting from single cell specific genetic modifications. And finally, this methodology can also be applied to a wide variety of disease models, including atherosclerosis, aneurysm, vascular ischemia in preclinical strategies. And all that's left is to thank my team members at the University of Cambridge, to the staff at VisualSonics for all their continued support. And thank you all for listening today. I'm happy to take questions, and do feel free to contact me on the below email address if you would like to. Thank you very much.
That's great. Thank you so much, Kirsty. That was an excellent presentation. So at this point, I'd like to open the floor to any questions. I see we do have some already streaming in, but if you have not had the chance to ask your questions, please use the Q&A button at the bottom of your Zoom panel. If we are not able to get to your question by the end of the session today, Kirsty has been very kind to say that we can directly refer you to her email so that you can have your question answered. All right, without further ado, we have lots here, so let's get going. The first is, how did you choose the ages that you studied? For example, the 44-week mark for the vascular decline. For example, did you test a time point such as 36 weeks in the mice?
So we just first, because what we saw a lot in the literature, that people were using very old mice and very young mice, and there wasn't a huge amount of data in between. So we wanted to focus on some of the time points that occur in the first year of the animal's age. So we just sort of picked them, not with any real, just we wanted something very young, middle age, slightly older. So we basically tried various different time points. We have done some measurements at 32 weeks, where we are beginning to see changes at that point as well. So yeah.
That's great, thank you. Another question here, do you ever image the carotid in M mode, or have you found value in using M mode in ultrasound?
Yes, we do B mode, M mode, EKV. We try to take as much data as we can from the animal.
Great. And what are your physiologic exclusion parameters for ultrasound evaluation, such as max or min blood pressure, breathing rate, temperature? And secondary to that, is there a trend as the animals age that makes this difficult to maintain across the cohort?
So things like, so we tried to aim for heart rates 550 above if possible. Temperature, we tried to keep between 37 degrees plus or minus 0.5 degrees Celsius. We keep isoflurane levels down to about 1.5%. Obviously, there's going to be different in different labs, but we try to keep parameters as much as possible within the ranges. We don't find this more difficult in the aging animals. We try to keep everything as consistent as possible so that we can make the comparisons as best we can.
Great. And the next question, did you measure both intracarotid and carotid femoral pulse wave velocity? And how do these values compare we find much lower values for IC in the ballpark of 1 meter per second versus CF, which is closer to 2.5 meters per second.
So just to check, you're asking the differences between pulse wave velocity being measured by invasive or non-invasive methods, is that correct?
I'm not sure. Whoever asked that question, maybe you could clarify a bit through the chat or the Q&A panel here. Yeah, I just have intracarotid compared to looking at carotid femoral pulse wave velocity. So I'm not sure if they're referring to internal or not, or invasive or not.
Yeah, sure, until they maybe write something. So I can say that we measure it the two ways that I described. So invasively with the blood pressure catheter, and also with the ultrasound non-invasively. And we've done a lot more invasive than non-invasive. But when we do compare them, we get very similar results, which is quite promising.
Okay, and I just got clarification. They're talking about non-invasively measuring. Non-invasively measuring intracarotid and carotid femoral pulse wave velocity and how those values differ.
Okay, so we don't do, we haven't done carotid femoral. We just do, invasively we do just in the aorta, between the 2 points in the aorta, or we've done also in non-invasive in the carotid. I guess the longer the distance between the 2 points, the better. There's obviously advantages and disadvantages to doing things invasively or non-invasively. So it depends on the requirements of your study. That's why we try to do both and try to maximize measurements for that as much as possible.
All right, great. And are there changes in Doppler blood velocity, or sorry, are changes in Doppler blood velocity and histology enough to prove presence of vascular stiffness in mouse models?
So, sorry, can you say that again?
Yeah, so they're asking, are changes in Doppler blood velocity and histology enough to prove the presence of vascular stiffness in mouse models?
I would say if you can do more, it would be better. I know that in the literature, they do accept the pulse wave velocity. They really look for that in terms of reviewers and things, because it is the gold standard. We also try to do lots of different parameters as much as we can, because it just enhances your evidence. Histology is very good as well. It can back your data up. But I would, if possible, if you can try to do a bit more than just Doppler blood velocity, it would strengthen your results.
Great, thank you. We do have a few comments in here that are just saying congratulations on the excellent presentation, the excellent work. And the next one here is, can you use this system to examine pulmonary vasculature for increased stiffness or fibrosis?
We haven't done that, but I would imagine that you could. I don't know how easy it is to image the pulmonary vasculature, but I would imagine that you could use this system for that. Yeah.
Yeah, so I'll just add in there as well. I think it would depend on which pulmonary vessels are of interest. You know, the lung can create artifacts that can make things a bit challenging, but if you're talking about the main pulmonary artery or any branches directly off the heart, that's absolutely possible. So the next question is, thank you for a clear and interesting presentation. I was wondering whether the nonlinear relationship between blood pressure and stiffness is an issue in these measurements.
We acknowledge that, but we just try to, we do need, often the parameters do require the pressure changes and the volume changes. And so we just basically try to standardize both components or both parts of the experiment, the imaging and the blood pressure. And then insert them into the formulae that we have for the different parameters of vascular stiffness. There's limitations with everything, unfortunately, but we just try to keep consistency in the way that we acquire the data and try to maximize the number of parameters that we get out so that we can increase our evidence for vascular stiffness. Yeah.
Excellent. So the next question is, was there a difference in survival regarding the different genotypes from week 44 to 72?
Not that we do see some sudden deaths in our mutant model. So this was the, if you remember, the vascular smooth muscle cell mutated form of OGG1, the inactive form. If anything, we do see probably more sudden deaths in those animals with age than the other strains. But generally, they all have quite a good survival rate, and we have aged them to 2 years in some cases, but generally we take them for experiments before that by 72 weeks, which is about a year and a half, before they start getting very, very ill and very sick and start having sudden deaths.
Excellent, thank you. And the next question is, did you manage to quantify how drastically the overexpressed mutation modifies survival? Was this more than wild type?
So can you clarify the question?
So I'll have to ask the individual who put that question in to maybe clarify a little bit more, if possible, and then we'll get back to that one.
Okay.
So the next question is pulse wave velocity equals distance over time. Do you have to measure Doppler in 2 different parts of the carotid?
So no, with the ultrasound system that we use with the Vivo 3100 and the Vivo in combination with the VisualVasc software, you only need to measure just using a single probe on the vessel, and then as long as you've got enough of a length from that one image, the software will allow you to take the 2 points from that same vessel.
All right, great. So another question, just confirming these data are all in carotid arteries. Are the changes to the stiffness mirrored in the left carotid? And secondly, have you examined the stiffness of larger vessels, for example, the aorta in this model? And thank you so much for a beautiful talk.
Oh, thank you. We use the carotid artery and the aorta mainly. We haven't looked at the left carotid. We tend to focus on the right carotid. So I don't have comparisons of left and right. We do see similar levels of vascular stiffness in the carotid and the aorta. We see it in both. We do our tissue measurements on aortic tissue mainly, imaging on the carotid, invasive measurements in the aorta.
Great, thank you. So another compliment, great talk. Whether heart rates affect do you find heart rates affect vascular stiffness measurements? We have just published a paper in which we showed that stiffness measurements using a fragment in the aorta is more sensitive than the one selecting aorta and femoral vein. Do you have a similar observation?
We try to keep our heart rates as consistent as possible within a range to try to control for that. We haven't looked at any correlations with heart rate and stiffness. We don't see any significant changes in heart rate between our groups, whether it's between the aged wild types or the our OGG1 strains. So we're confident that the changes we're seeing are not related to heart rate.
Excellent. Can compliance still be measured after vascular injury?
Not sure about that one. We don't work with a vascular injury model, but I would I'm not sure on that one. I'd have to come back to you on that one.
Okay, that's great. And I noticed that was an anonymous attendee that inputted that. So if you'd like Kirsty if you'd like to discuss that further, please comment with your contact information. So the next question is, again, starts with a nice compliment. Thank you for a lovely talk. How easy is it to measure vascular aging in mice that also have atherosclerotic plaques?
Good question. I don't have experience with that, I'm afraid. I haven't imaged animals with atherosclerotic plaques. I imagine it would still be possible, and yeah, I'm afraid I can't comment further as I haven't got experience in that.
Well, thank you. So the next question is very nice presentation, another nice compliment. And I was wondering just what the sex of the Black 6 mice you studied was.
We used both male and female. We didn't want to bias between a single gender, and we didn't see differences between the two sexes, so we used both.
It's very interesting. So the next one is, what are the signs of aging in microvasculature, for example, in capillaries?
That's a good question. A lot of the data in the literature is focusing on large arteries. I would need to come back to you on the precise changes in the capillaries.
Okay, that's great. Forming lots of collaborations here. So another question came in, is the carotid preferred over the aorta for measurements of pulse wave velocity?
Actually, I think the aorta would be better, only because it's a larger vessel, and the larger the distance between the 2 points that you're measuring, the better, the more accurate the data. Carotid is perhaps slightly easier to image because you don't have other organs and tissue in the way. It's a bit more isolated. So it's just really about what your experience level with imaging is. And but I think the aorta is probably the preferred, just because it is a larger vessel.
Great, thank you. So the next one says, thanks for the nice presentation. Did you look at the heart function in addition to the pulse wave velocity measurements?
No, we didn't. We don't do cardiac measurements at the moment, just focusing on the vasculature.
Excellent, thank you. So another compliment here. Thank you very much for the nice talk. Did you investigate any structural changes in your animals? If yes, what kind of changes did you see?
So we do look at structural changes. We, as I showed in the talk, we do look at things like collagen levels and elastin breaks histologically. We also, what we've also done in our most recent study on the OGG1 strains is an proteomics screen of extracellular matrix proteins that are changing with age, changing with oxidative stress. So I haven't shown that data today, but we're looking into that, and we see lots of changes in different extracellular matrix proteins that are of interest to us. And so we're following that up. But yeah, we do try to supplement our data with structural data as well.
Great, thank you. So this is maybe some thoughts for future experiments, but have you tried collecting 4D data from the carotids? There could be a way to estimate pulse wave velocity from 4D data or four-dimensional data.
No, we haven't, but that's a very good suggestion and something we'll look into. Thank you.
Next question, we're really grilling you here. There's lots of interest. Thank you very much for the nice presentation. Is there a possibility to estimate change in pressure non-invasively with ultrasound?
So can you repeat the question?
Is there a possibility to estimate change in pressure non-invasively with ultrasound?
Yes, there is options to do that, but I would recommend doing it invasively. I think invasive, although it comes with its disadvantages, and it would usually involve a terminal procedure to the animal. I think that it's more accurate to have intra-arterial invasive measurements of blood pressure than relying on blood pressure cuffs and things like that, but it is still possible.
Great, thank you. So nice talk, thank you so much. Did you check for some correlation between pulse wave velocity and molecular mechanism activation, for example, pro-fibrotic pathways like TGF-beta or specific extracellular matrix protein secretion?
Yes, that's what we're working on at the moment. We haven't fully done all the correlations, but we're looking to make those links where we can.
Great, thank you. I'm just anticipating your next moves there. I'm just sifting through these questions. So someone, this is more about our technology. I'm still on the Vivo 2100 and don't see the software. Is the EKV 3100 feature or inside Vivo Vasc Vivo strain? This might be an application question rather than a presenter question. It kind of is, so I'll field that. And yeah, EKV is available both on the Vivo 2100 and 3100, and we can follow up with you later with more information on that offline. So another question was, do you measure Doppler blood velocity from the carotid artery as well?
We do, yes.
All right. Sorry, there's a lot of questions I'm sifting through here. For your anesthesia, are you using medical air or carbogen 90% O2?
I think it's medical air.
Okay, great. What is your suggestion for combining parameters used non-invasively to stratify age groups?
I'm not sure I follow the question.
Okay, well, we can always connect you two after, and you can elaborate a little more perhaps on the question. Would you be so kind to elaborate more on 3D reconstructions of vessels? Which results you could quantify with 3D?
So I don't have too much experience on the 3D reconstruction. I wouldn't be able to comment much on that, I'm afraid. Yeah, sorry.
Yeah, no problem. I can comment on 3D reconstruction. It is absolutely possible with our system to do 3D reconstruction on any vessel you can really see. And again, we can follow up, our team can follow up with you after on what specific vessels you were interested in and provide you any tips related to imaging those. Let's see. Was dynamic compliance tested or challenged with a dilator or constrictor?
No, it wasn't, but that is something that we're looking into trying.
Great. So we have been asking you lots of questions, so I'm just going to do a couple more, and then we will have to follow up with everyone else after the fact. But I do encourage you, please continue to put in your questions, and we will make sure you're connected with Kirsty after the fact. So another more compliance-related question: how do you calculate compliance and distensibility non-invasively? Do you see differences in these metrics compared to stiffness?
We use, as I said, a mixture of non-invasive and invasive measurements to measure our compliance and distensibility. So we've obviously got the imaging data, which allows us to drive diameters, and then we get blood pressure measurements invasively, and the two are slotted into the formulas that I outlined in the presentation. There are ways to measure distensibility using the Vivo Vasc software. I believe you'd still need pressure data, but maybe Christina can confirm that.
Yeah, we can follow up with that as well, and answer really more in-depth questions with that individual after the fact. So we can get to maybe one more question, and then I think we'll cap it off for the day. So the next question is, thank you for your excellent talk. You said in order to measure pulse wave velocity non-invasively, you need a vascular segment that is long enough. Can you please specify the length that is needed, or that that you set as your minimum?
The longer the better, really. You know, a carotid is just millimeters long. It's probably better, the longer the length you can use, the better, the more accurate the measurements. We don't really have a limit because we also do this invasively, and we're just still kind of exploring the non-invasive method for doing this, but I would always suggest to use as long a vessel length as you can.
Great. Thank you so much, Kirsty. Thanks so much, everyone, for joining us today. Thank you for the excellent presentation. It obviously spurred a lot of interest and maybe potential collaborations in the future. So I do want to thank you so much. Again, we will be sure to follow up with everyone whose question we were not able to get to today. And if you weren't able to input your question, or you still had it top of mind, please contact us through our resource portal or our website, and submit any questions you might have, and we will be sure to contact you and get back to you right away if you request a question through a web form. You can also check back on our upcoming webinars or our past webinars by accessing it on our website. You can find us on all the major social media channels, and you can visit us at many virtual conferences that are upcoming. And then finally, for anyone who has some more technical questions, we do have an online training resource portal called Learning Hub. So please feel free to check that out. Again, thank you so much for joining us today on this lovely December day. I want to thank you all so much and wish you very happy holidays on behalf of all of VisualSonics. And Kirsty, thank you again so much, and we'll talk to you all soon.
Thank you very much.